Very good evening to one and all. I welcome you all for the Reda Academy. This is my first live session on asexual and sexual reproduction in plants, the chapter 1 from Botany. My name is Dr. J. Devi, and today I'm going to give you a detailed view on how plants are reproducing. So, the word reproductions is very familiar to you all that you had studied in your previous classes. The word reproduction refers to the process by which organisms are producing their own kinds. Re means again, and production means is a process of producing their own kinds. So organisms are producing their own kinds. For example, a bacteria that give birth to a daughter cells or a cat that give birth to its own kittens are called as the reproduction. So this reproduction is not a life process, but it is a vital process for the extinction of species. If a species has to survive on earth, then it has to reproduce. So there are a lot of changes that happens during the time of reproduction. The variation is one that is very important as it is leaving a path for the evolution. So the offspring which is produced during the reproduction is not an identical clone of their parents. There are some difference seen in the offspring which is needed for their survival on the earth. So variation is called as the key for the evolution. This reproduction, especially if you are taking it in case of plants, is important not only for the survival and also it is needed for the continuation and the existence of all our organisms. And it is little harder for you to understand how the plants are getting into reproduction. Today, I'm going to make it very easier for you to understand it to the core. When talking on the types of reproduction, there are two different types of reproduction. One is asexual type of reproduction, another one is the sexual type of reproduction. And in your previous classes, you may heard about these two terms and you studied what is the difference between these two type of reproduction. The first one let we discuss first, that is asexual type of reproduction. In the asexual type of reproduction, only one parent is going to involve in it. That is, single parents are going to involve in this type of reproduction. So single parent, so there is no distinct male and female present in this organism. Especially lower plants are involved in the asexual type of reproduction. So single parents are going to involve in the asexual reproduction. Here you cannot identify male, female and male gamete, female gamete. So these concepts are not present in case of a asexual reproduction. And as there is no formation of gamete, there will be no fusion of gamete. And there is no a step called as fertilization is absent in case of a asexual reproduction. So how this is going to multiply? A very simple concept of cell division, that is mitosis. So the name of the cell division is called as a mitosis. So I just want to give you a brief on this mitosis. So let me have a look on mitosis. The word mitosis, I'll just show you that. So mitosis, this is one type of cell division. Here, if you take a cell contain nucleus in them and they have diploid number of chromosomes. For example, if you take a human cell, they contain 46 number of chromosomes. So this 46 number of chromosome, during the cell division, they will going to form two cells, and that two cells is going to contain nucleus in them. That nucleus will also contain 46 chromosomes. So this is called as a mitotic cell division. So here you couldn't able to see any change in the number of chromosomes. Okay, but if you take meiotic cell division, if you take a meiotic cell division, here if you take a cell that contain a nucleus and that have 46 chromosome in them, but during the meiotic process, this 46 chromosome will be 23. So there is a reduction. That's why it is called as a reduction division. You are calling it as a reduction division. Okay. So this meiotic cell division always happens in case of a gamete cell. So it may be a male gamete or it may be a female gamete. That is in case of a sperm, and in case of a ovum, you could see only haploid number of chromosome. Haploid number, that is represented by the letter N, that is 23 chromosome is present in case of a gamete. So during the gametogenesis, that is formation of sperm or ovum, this 46 number of chromosome will divide into 23, and this happens only in the gametes. Whereas if you take in case of a mitotic cell division, here this happens in all the normal stomatic cell. So if you take a stomatic cell, that is a normal cell which present in all parts of your body, in that case mitotic cell division will take place. So meiotic cell division that happens in the gamete, and that is going to reduce the number of chromosome into half, whereas the mitotic cell division, here they are going to just divide as such. So that is the two types of cell division. So asexual reproduction is one which is going to show you the same number of chromosome in their offspring. So asexual reproduction prefers mitotic cell division, and individuals which is going to form as a result of this asexual reproduction are genetically identical. They are just like a clones. They are just like a clones. So clones means the exact Xerox copy of their parents. So they are genetically identical as their parents. So you couldn't see any variation happening in case of a asexual type of reproduction. Coming to sexual type of reproduction, here two parents are involved in the reproduction, a male and a female. Male and female can be easily distinguished by their morphological features, by their anatomical feature. So male and female are very distinct in case of a sexual reproduction, and the male is going to produce the male gamete, a female is going to produce the female gamete. So gametes are going to produce and they are going to fuse together, and there is a formation of zygote by the process called as the fertilization. Fusion of male and female to produce the zygote is called as a fertilization, that is going to happen in case of a sexual type of reproduction. And here the very primary process of cell division is the meiotic cell division, that is a reduction type of cell division. The 46 number of chromosome is going to break into 2F, that is 23 chromosomes are going to be there in their offspring. And later there are some conditions where they go for mitotic cell division, that is after the formation of zygote, then they will be going for the mitotic cell division. So male and female is going to form by meiotic cell division. After the fusion and zygote formation, they will be going for a normal type of cell division. That is, the zygote will be divided into two cells. So that is a sort of mitotic cell division. So meiotic is the major type of cell division, and after the zygote formation, they get into the mitotic type of cell division. So individual shows variation here because two different type of chromosomes are going to involve, that is of male and female. So you can expect variations in the sexual type of reproduction. Then, as I said, the mode of reproduction is of two type, asexual and sexual type of reproduction. This is very commonly, I just given you the difference between these two. When you're talking on plants specifically, here the asexual reproduction, you may wonder about the asexual reproduction in plants because we studied about the different types of asexual reproduction and all other organism. In a general, we had a point like this. So this asexual reproduction, here you can see fission we studied. In case of a bacteria, that is one cell is divided into two, that is called as binary fission, and sometimes the one cell is divided into many, that is called as multiple fission, that you heard in your previous classes. And budding, very aware about budding in case of hydra, that is in a hydra. So here the small bud will be formed and they will be detached to form as a new hydra. So this is a type of asexual reproduction you study. And fragmentation, that is in case of a spirogyra, one cell is being divided into many fragments and each fragment can give rise to a new organism. So then gemma formation, regeneration, sporulation. So those are many different types of asexual reproduction that we are aware about. When you're talking on asexual reproduction, which is narrowed down to only plants, in that case, the asexual reproduction will happens in a plant which is happening only in the vegetative parts are going to participate in the formation of a new organism. So there you cannot expect any seed formation, there you cannot expect any spore formation, and flower is not involved in this type of reproduction. So in the asexual reproduction, there is no involvement of the flower because flower is called as the sexual reproductive part of a plant. So flower is called as the reproductive part of the plant. So it is not going to involve in the asexual type of reproduction. So what is going to involve in the asexual type of reproduction? As I stated, it is the vegetative part is going to involve in it. Either may be a leaf, it may be a stem, or it may be a root. So a new plant is going to arise from the vegetative part. It may be a leaf, it may be a stem, or it may be a root. But if you take a sexual reproduction in a plants, then a new plant will arise from the reproductive part, that is flower. So flower is going to participate in the sexual reproduction of a plant. You are clear? So okay, fine. So what is the product? What is being formed here is the fruits and the seeds are going to be the product of the sexual reproduction. So that's a difference between the asexual and sexual when you are concerning about the plants. If it is happening in a plant, so there is a variation. So this lesson is going to talk on both the type of reproduction, that is asexual as well as sexual type of reproduction. So in this session 1, I'm going to discuss in detail about the asexual type of reproduction that is going to happen in case of a plant. So chapter 1 we are generally going to talk on reproduction in plant. So two type of reproduction as I stated, asexual and sexual. We saw what is the difference between asexual and sexual, and we just narrowed down the topic to the uh, plants asexual and sexual reproduction. And now we are getting into the core of the lesson. The first half of the lesson is going to talk on the asexual type of reproduction. So in that asexual type of reproduction, we are going to totally talk on the vegetative propagation. As I stated, either root, plant or the stem is going to involve in the formation of new plant, that is called as a vegetative reproduction. So flower is called as the reproductive part, whereas the buds that is grows from a leaf, stem or root is called as the vegetative part. So here the vegetative part, as I stated, root, stem and leaf. So the buds are going to arise from these organ, and the buds are going to grow, and they will be growing as a new organism. That is, new plants are going to arise from the parts that are raised from the vegetative parts like root, stem and leaf. Sometimes these buds, that is which is being arise from the root, stem and leaf, are going to act as a organ of storage. So they will be storing starch in them, and they will be act as a organs of storage. So that is the a beautiful special modification of reproduction. Flower is the reproductive part, and androecium and gynoecium are the unit of the reproduction. But if you take a vegetative reproduction, here the reproductive propagules or the diaspores are called as units of reproductive structure. It is being used for the process of propagation. I'll give you one thing to understand this reproductive propagules. If you take a potato, in a potato you can able to see the eye region like this. So here this is called as a reproductive propagule, which is going to form like a bud, and a new plant will arise from the eyes. So these are called as the reproductive propagules or diaspores. So which is being the main reason for the formation of new plant. So I'll just give you an outline about what are all the topics we are going to cover in this session. So this vegetative propagation, we are classifying them into two. One is called as the natural method, another one is the artificial vegetative propagation. Natural vegetative propagation is being carried by the nature, and humans are not involved in this. So they are going to choose either root, stem, leaf or the bulbil, that is the storage organ, for the formation of the next generation. It may be a root as I stated, or it may be stem. If it is in stem case, you are having three different types of stem which is going to participate in the natural vegetative propagation: underground, subberial and aerial. Underground, I think you know, it is under the soil, and subberial is the stem which is present above the soil. So these are all the varieties: rhizome, tuber, bulb, corm, sucker. So these are the five different varieties which is coming under the case of a underground stem. But if you're taking a subaerial stem that is seen above the ground, then you are having runner, offset and stolen. So runner, offset, stolen. So these three are going to come under the subaerial type of stem. And if you're talking about the artificial vegetative propagation, here humans are going to do this vegetative propagation method, and we are going to do all the process to just extend it from the agriculture, just to get a good yield, good amount of yield, and just to propagate them, we are going to do it artificially. Under this artificial vegetative propagation method, we have cutting, layering, grafting, approach grafting and micropropagation, which is an advanced technique, that is modern vegetative propagation. Let me see one by one in detail to get you a great idea on how the plants are getting into vegetative propagation method. So, first we are going to see on asexual type of reproduction. In that we are going to see on vegetative propagation. Under the vegetative propagation, we are going to see the first division, that is the natural method of vegetative propagation. So, natural vegetative method of propagation, as I stated, it is being done by the vegetative parts, that is root, stem and leaf. First, we are going to see on the root. So, when talking on the root, as I stated, root is going to be present under the ground and they are going to develop as a new plant, that is either the vegetative or the adventitious bud that is present on the root is going to grow as a new plant. So, some example I stated here. One is the murray. This murray is actually the Tamil you are calling it as kariveppile. Okay, so that murray is one good example of the vegetative natural vegetative propagation done by the roots. Okay, and the second one is the uh, Millingtonia. In Tamil it is called as தேன் பூ or குழல் பூ அப்படின்னு சொல்லுவாங்க இத. So this Millingtonia is another example of natural vegetative propagation that is done by the roots. And the third one is called as the Dalbergia. This is actually a tree. These three different types of plants are taking roots as their method of vegetative propagation. Are you all clear? Is it clear? Fine. Then let me go to the second example of vegetative reproduction in roots. Some tuberous adventitious root. And I think you are aware about these two types of root. One is called as the adventitious root, another one is the main root. So that is a root that is being formed from the radical is called as the, just a minute, I'll tell you what is that. Yeah, fine. So I'll draw you the picture of root. So this is, okay, this is the structure of a root. So this is called as a primary root, and this is called as the secondary root, and the one which is formed from here is called as the tertiary root. This is the structure of a taproot. Okay, and if you take in case of a fibrous root, here you cannot see any primary roots, so there will be a bunch of secondary root, and tertiary roots will arise from here. Okay, so this is a fibrous root. So both the case of a taproot and a fibrous root, during the process of seed germination. Yes, yeah. So during the seed germinates, you have radical and you have plumule. So this is called as a plumule and this is called as the radical, this lower portion. Okay, if the root arises from this radical, then it may be of two type. One is called as a taproot, another one is called as the fibrous root. And this plumule is going to be the shoot system. Suppose if a root that does not arise from the taproot, it is called as the adventitious root. So, these adventitious roots will grow either from the branches or from the sides of the stem. So, this is going to give you additional support to the plant. So, such a type of root is called as the adventitious root. So, you are clear on the adventitious root? So, adventitious roots are going to give you additional support, and that is not going to form from the uh, main radical. Okay, so that adventitious root, apart from the developing bud, they are not only involving in the vegetative propagation of producing buds, and they're also involved in the storage of food. Okay, so they are going to act like the vegetables. Here I stated some of the examples of this roots, storage root. One is called as the Ipomia baptis, the very well known example, sweet potato, Chakravalli kadam. Okay, and the second one example is a dahlia. Here I had given the what you're calling it as a tuber, that is kerangu. Okay, from this only you are getting dahlia plant. So the one ornamental plant is the dahlia. Okay, so this dahlia has the tuber in them. From this they are going to form. So both these are example of a tuberous adventitious root, which is also acting like a storage and also helpful for the vegetative propagation. So this is the example. So we have two example. One is the murray, which is called as Caryophyllae, and Millingtonia, which is called as Kuralpoo, and Dalbergia. These are all the example of vegetative propagation by root. And some adventitious roots are going to act as a storage. One is the Ipomia baptis, which is called a sweet potato, Chakravalli Karanga, and Dahlia, which is also propagate using the root. So natural vegetative propagation method, here roots are going to act as a organ which is going to produce a new organism. ஸோ ஒரு புது பிளான்ட் வளர்றதுக்கு இங்கே ரூட்டு தான் நம்ம ட்ரான்ஸ்ஃபர் பண்ண போகிறோம். ஸோ நேச்சர் இஸ் டூயிங் இட் ஆன் இட்ஸ் ஓன். ஃபைன். அண்ட் த செகண்ட் டைப் ஆஃப் வெஜிடேட்டிவ் ரீப்ரொடக்ஷன் இஸ் தி த்ரூ த ஸ்டெம். Here stem is going to act as a vegetative part. So aerial stem modification, underground stem modification, subaerial stem modification are the three different types of stem modification which is going to helpful for the vegetative propagation, and nature is doing it on its own. We are not involving in it. And in that first let me talk about the underground stem. As I told you, this stem is going to just present below the soil. Sometimes you may have a little confusion whether it is a stem or it is a root. So soil, so below the soil, the stem will be present, and sometime they will be storing food. And this is called as the underground stem. And how could you identify this underground stem? You couldn't able to see any root structures here. If no root structure is present on that, then that is called as an underground stem. So I stated 4 examples for the underground stem. One is the stem tuber. Best example is the potato. So the eye which is present in the potato, so this is a region called as a eye. So this region is going to form a new plants. So that is stem tuber. We are calling it a stem tuber. And second one is the rhizome. Here ginger is the example. Ginger is the example for rhizome. So they are going to give rise to the new plants. And the third one is the bulbil. Here onion is the example of bulbil. And idu neenga veetla paathirupinga nenikra. So onion, if you are keeping it for a very long time, it will start growing the plants. So onion is an example for the bulbil, which just called as an underground stem, which will lead to the formation of a new plant. And the third one is called as a corm. Corm is actually the colocasia is an example of corm. Chepakarang, you are calling it as chepakarang, right? So that is an example of a corm. So all these four, tuber, rhizome, corm, bulbil, are the example of underground stem. Okay, and if you are taking subberial stem, that is stem that is seen above the ground, and they have the root, and some of the stem are just above the soil. So it is just above the soil, and you can feel just like a root, because soil, if it is like this, அப்படியே சாயில் ஒட்டியே தான் அந்த ஸ்டெம் இருக்கும். So that is called as subaerial stem. So we have four different types of subaerial stem which is acting like a vegetative propagators. One is the runner. Example is the strawberry. I showed you the picture here. So one mother plant will be here, and this they are having a sub root, everything. A mother plant will be here. Then the subaerial stem will grow above the, just grow above the soil. At one point they have a node, and that node will give rise to a baby plant, baby strawberry plant. Okay, and after a period of time, this subaerial stem may break, or the network will follow to the other baby plant from this. So like that, they will just look like a network. You can't able to separate one plant at a time. So you can see the bunches of them for a wide area. They just formed like a group. So that is called as a runner. Strawberry is an example for it. And if you take stolen, mint is a good example of stolen. Here you can see them. So they are having a group, a bunch of plants are being raised up from one single root. A bunch will be there, and a stem will be connecting all these two. Okay, but that bunch will happen only in a group. So after that, one more group will be formed. So that is called as a stolon. And here in case of a sucker, chrysanthemum, samandip, that is an example of sucker. Here you could see that small plants are being arised from the mother stem, and that will be growing individually as a new plant. And offset, this is actually you can use to see this pistia plant in case of water. This is actually an aquatic plant. So here you can see that stem is just acting as a vegetative propagator. From the mother plant, they are just arising up as a individual small baby plants are being formed. So through the stem, they are extending their propagation, and that's an example of a subaerial type of stem. So the third one, the very well known example we studied in all our classes, is the vegetative reproduction by leaves. The well known example is the bryophyllum. Here at the margin of the leaf. So at the margin of the leaf, here at the margin of the leaf, a small buds, adventitious buds are being formed up, and they are forming roots. Then they will fall off and they grow as a new plant. So through the leaf, they are doing their vegetative reproduction. So until now, we see about the three different types of natural vegetative propagules. One is the root, another one is stem, and third one is the leaves. So what is the advantage and disadvantage of natural vegetative propagation method? First, let me take what are all the advantages of it. So as they are doing through their vegetative parts, they no need any external agencies to do the process of pollination. And I think you are aware about sexual reproduction. In plants, if you are taking the sexual reproduction, if you are taking in case of a plant, as I stated, the flower is the reproductive part. In case of a flower, if you are taking flower, the pollen grains should be travel from, it has to travel from the anther to the stigma. I just draw you one picture. So flower, they are having the filaments which contain the anther, and anther have their pollen grains. So these are called as the pollen grains. So this pollen grains has to transfer from anther to stigma. Stigma will be the male part, that is a gynoecium. On that, the pollen will be deposited. So this process is called as the pollination. You're calling it as pollination. So pollination can be done either by abiotic factors or by the biotic factor. If it is an abiotic factor, either water or air will do it. But if you take in case of a biotic, insects, birds are going to do this process. So pollinators, they are called as pollinators. So a plant has to depend on these pollinators for doing the process called as the pollination. But if you are taking this vegetative propagation method, you need not to expect, that is a plant need not to expect or need not to develop the nectar gland to present the nectar to the insects, or they should not be colorful, and they no need to develop some evolutionary changes like modifying their stigma, and that are all are not needed because they no need any external agencies to do this pollination. So they can do it just like propagating through stem leaf for them. So insects, abiotic or biotic, nothing is needed for them. That is a good advantage actually. And because they have to offer something for them, and the chance of pollination will be very less in case if they are preferring such pollinators. But it was easy method. So just like that they can do it. So food is usually present in the vegetative structure, and that will lead to the rapid growth of the buds into a daughter plants. As I stated, the buds are not only doing the vegetative propagation, but also they are storing the food material. With that food material, the daughter plants are just growing just in a happier way. But if you take a seed that have a very limited amount of food material in them, they have very limited time. Within that, they have to germinate, and then they have to go for photosynthesis to just develop them. But in case of this vegetative propagation method, you are having enough amount of it for the daughter cells to grow. So it's an advantage. And the third advantage is that new plants resembles the parent plant in every way. Just they're going to be just like a clones of a parent. Clones of a parent. Exactly what you are required, that will be projected by the nature through their offspring. So only one parent are involved. There is no struggle for the gamete formation. Lot of energy being spent because of the gamete formation that can be saved, conserved by the plants. And they are just spend that energy in some other process. So only one parent are involved, and there is a chance of giving up lots and lots of other conservative process using that energy. So these are all some of the advantages of the natural vegetative propagation. What is the disadvantage? What nature is setting as a disadvantage? So lack of dispersal mechanism may lead to overcrowding. If the plant is not, if the seed is dispersing by animals, for example, இப்போ மாடோ இல்லை ஏதோ ஒன்று வந்து சாப்பிடுது அப்படின்னா, so it will go and disperse them in a very longer, wider area, and the species will take a very longer area to survive. Enough amount of sunlight கிடைக்கும், water கிடைக்கும், எல்லாமே கிடைக்கும் if it is being dispersed in a very larger area. But as it is doing by the natural vegetative propagation method, அப்படியே பக்கத்து பக்கத்துலேயே அந்த plants எல்லாம் வளர்ந்துடும். So there will be overcrowding happens in case of a vegetative natural vegetative propagation method. So that is one disadvantage obviously. And new plants are less varied. So as they are clones, you cannot expect any variations in them. அப்படி parent மாதிரியே தான் அது இருக்கும். If suppose the parent is having any genetical disorders, அதுக்கு ஏதாவது genetical disorders இருந்துச்சு, ஏதாவது error இருந்துச்சு, the chromosome, that will be traveled to the offspring. That is one a great disadvantage actually. And the new plant may be less adaptable to the environmental changes. So because variation is not happening, so you cannot expect them to be adapted to the environment. So உங்க parent என்ன மாதிரி ஒரு habitatல இருந்தாங்களோ அதே habitatல தான் அந்த offspring இருக்கும். So it is one disadvantage. So you cannot expect these plants to be cultivated everywhere because they are all just clones. So they will also follow the same habitat. I just given you a picture of a sniffers, a type of a grass that is being crowded, overcrowded in an area because of this natural vegetative propagation method. So I think you get a good idea about natural vegetative propagation method. Nature is setting its own propagation using root, stem and leaf. They are just expanding and rising up their own species. And now humans are always having a good curiosity in making things on their own. நம்மளே அதை செஞ்சு பார்க்கணும் அப்படிங்கிற ஒரு curiosity humans எப்பவுமே இருக்கும். Right? So we just understand that natural vegetative propagation, and we studied how the nature is doing it, and we are taking it on our own. So because nature வந்து எப்பவுமே ஒரு விஷயத்தை ரொம்ப slow and steady தான் பண்ணும், கரெக்டா? So but humans have no time for all this because we have to offer food for a very larger scale of population. So agriculture பண்ணணும், horticulture பண்ணணும்னா we cannot expect for the nature to take such a very long time to do it. So humans will always have running out of time. So they are taking this natural method on their own in their hands, and that is called as the artificial propagation. Here also we are using only stem, root and leaf. But we are doing it on our own. Nature is not doing, but we are using as nature is doing, but in a larger scale as a human. That is the difference between the natural vegetative propagation and artificial vegetative propagation. There are two methods in this type of artificial propagation. One is the conventional method that man is being using this for a longer period of time. நம்ம பழங்காலத்துல இருந்து அப்படியே பண்ணிட்டு இருக்காரு. என்னன்னு கேட்டோம் அப்படின்னா, ஒன் எக்ஸாம்பிள் ஐ கேன் ஸ்டேட் யூ. இப்போ முருங்கை மரம் நம்ம வீட்டில் வைக்கணும்னா என்ன பண்ணுவோம்? ஒரு முருங்கை கிளையை வந்து வேற ஒரு வீட்டில இருந்து வாங்கிட்டு வந்து வைப்போம், கரெக்டா? So that is one called as a conventional method. Here that doesn't need any technical knowledge and all. We should know only one thing. Is it a stem propagation, root propagation, or stem leaf propagation? That's it. அது போதும், அந்த knowledge போதும். So we can cut that part, put it up, and we have to provide a proper environment for them, give them a good amount of nutrition, water supply, and they will grow. So this is called as the conventional method of vegetative propagation that is done by humans artificially for a very longer period of time. But when you're taking a modern method, here you are using technologies like tissue culture, micropropagation, and we are doing it in laboratory condition. Highly technical knowledge is needed, and but here what is the greater advantage is that you can do n number of offspring in a very short duration of time with this technical knowledge. So large number can be produced in a shorter period of time. This is always human desire to do it. So we are doing it, and that technique and that method is called as the modern method. We are going to see both of it under the artificial propagation. So the first one is the conventional method. As I stated, very long duration of time humans are following them. So three things we are going to discuss here: cutting, grafting, layering. The first one is the cutting. So cutting either the root, stem or leaf of the parent, and we are going to place it in a suitable medium to grow, that is a soil, and we have to provide them what are all the necessary things. So proper sunlight irukkan paakano, correct adukku watering, irrigation kudukano, then and the soil quantity of nutrients that's it. Then they will be started growing. So there are three different cuttings as I stated. One is a root cutting, stem cutting, leaf cutting. So stem cutting is widely we are using for mostly of all type of plants. So root cutting, if you are taking malice is an example for the root cutting. ஆப்பிள் தான் வந்து malice. So ஆப்பிளோட plants எப்படி propagate பண்றாங்கன்னா only through the root. So they will be cut a part of a root, put it in a soil, grow it, let them form the plantlet, then they will be transferring this root to the field. நிலத்துக்கு மாத்திடுவாங்க. Then like the stem, as I stated, முருங்கை மரம். So stem cutting, we just cut the stem and grow it up. And bryophyllum, the good example is a leaf. ஒரு leaf இருந்தா போதும், you can grow this bryophyllum in a very larger scale. So these are all just example for cutting. Either root, stem or leaf has been used for the process of artificial vegetative propagation. And the third one is called as a grafting. Here we want to make some variation. We want to combine two different plants together, and we just want to do some experiment by just mixing up two different types of plants. So what you do, you go for the method called as a grafting. As I stated, we are taking two plants. One is going to act as a stalk, another going to act as a scion. So one is going to act as a stalk, the plant which is having contact with the soil. So in the plant to the root soil irko, that is called as a stalk. And in the plant to the root soil illa, but plant to the stem root la irukadodukku mela nam vechomna, that is called as a scion. So scion has no attachment with the soil, stalk has attachment with the soil. So this is a good picture which shows you. Here it is the stalk which is having the root at the soil, and you are placing a stem over it, and that is going to act as a scion. The best example for this grafting method is mango. So mango la vandu ottu manga appdin solluvaanga. And the word neenga kelipattirupinga nenikiren. So we have nearly hundreds varieties of mango. So ella mango it's just a combination of two different varieties. So sweetness kaga, juice kaga, and very romba thinnana layer skin irukra madri, nariya fleshy region irukra mari, naar kammiya irukra mari, and the mari we are just doing it between two different mango varieties by the technique called as a grafting. And we have 5 different type of grafting method. ரொம்ப காமனாக இருக்கிற விஷயம் ஸ்டாக்கு சைஸான், அதில் எந்த சேஞ்சும் இல்லை. எப்படி அந்த ஸ்டாக் அண்ட் சைஸான நம்ம ஒன்றா சேர்க்குறோன்றதை வச்சு தான் we are just classifying it. But if you are taking first method called as a bud grafting, in the bud grafting method, you are just going to put a T-shaped incision. ஒரு சின்ன கட் மாதிரி T ஷேப்பில் ஒரு கட் ரெடி பண்ண போகிறீங்க. அந்த கட்டில் what you are going to do, you are going to put the size on, that is bud. அந்த bud ஒரு லீஃபாக இருக்கலாம், இல்லை ஒரு குட்டி ஸ்டெம்மாக இருக்கலாம். You are just going to put it. The bud is just a growing part, அவ்வளோதான். அது ஸ்டெம்மோட growing பார்ட்டாக இருக்கலாம், இல்லை லீஃபோட growing பார்ட்டாக இருக்கலாம். So that bud will be attached on the T section, like insert பண்ணிட்டு, then you have to tie it up using the ஒரு மாதிரி பாலித்தீன் கவர் மாதிரி இருக்கும், which has been mainly used only for the purpose of graftingகாகவே இந்த கவர் ஒரு மாதிரி யூஸ் பண்ணுவாங்க. ஸோ அதை வச்சு நம்ம என்ன பண்ண போகிறோம், we are going to tie it up, and after that a new plant will arise from this region. ஸோ இதை வந்து நீங்கள் வீட்டில் செஞ்சு பார்க்கலாம். I just tried one thing. நான் ஒன்று ட்ரை பண்ணேன், இந்த வெஜ் கிராஃப்டிங் நான் வீட்டில் ட்ரை பண்ணியிருக்கேன். ஓகேவா? ஸோ the second one is called as approach grafting. இது கொஞ்சம் டிஃப்ரெண்ட்டாக இருக்கும் யோசிக்கவே. ஒய்? பிகாஸ் யூ ஆர் ஹேவிங் அ டூ டிஃப்ரெண்ட் பிளான்ட்ஸ். ஒன்று வந்து பாட்டட் பிளான்ட், இன்னொன்று வந்து நார்மல் பிளான்ட் விச் இஸ் ஹேவிங் ரூட் இன் தி கிரவுண்ட் இட் செல்ஃப். அந்த ஸ்டெம்மையும் வி ஜஸ்ட் மேக்கிங் சம் ஸ்க்ராச்சிங், லைட்டாக அந்த ஸ்டெம்மை வந்து ஸ்க்ராச் பண்ணிவிட்டு வி ஆர் ஜஸ்ட் joining it together and tying it up. A new branch was developed which is having both the character. I was really wondered when I see this in a horticulture platform. So horticulture unit ku poitu idu naanga live ah paathom approach grafting. And third one is called as the tongue grafting. Here you are making an incision like a tongue. Naak madri oru incision ready pannittu. We are just adding two different plants together. And the fourth one is a crown grafting. This is also one important type of grafting done in uh, trees especially. So trees la vandu we are just cutting up the trunk and we will be placing 2 to 3 different branches of other type of tree and we are making it like a crown. So crown la irukra ma indha madri attach pannidum. So that is called as a crown grafting. And wedge, as I said, wedge madri V shaped we are cutting up and we are inserting a new plant on it and they will be growing up. So it's a combination of two different plants together. So that is called as a wedge grafting or grafting. 5 different varieties are there. And the third one is called as a layering. Here, stem of the parent plant is allowed to develop root while still intact. ஒன்றுமே பண்ண போகிறதில்ல. ஒரு பிரான்ச்சை மட்டும் எடுத்து சாயிலில் வச்சு அதுக்கு மேலே ஒரு வெயிட் போட்டிங்கன்னா what happened? The stem will have a contact with the soil and they start rooting there. Simple. இது நம்ம நிறைய ரோஸ் அண்ட் ஜாஸ்மின் பிளான்ட்லலாம் இது நம்ம ட்ரை பண்ணியிருப்போம். So the root is going to just form the region where the branch is going to have a contact with the soil. Okay, instead of cutting and doing, we are just placing it on the soil. After the branch is going to form a root, then we are going to cut the part. So இப்போ இந்த இடத்துல வந்து இந்த மாதிரி root form ஆனதுக்கு அப்புறமா we will be cutting this plant alone and we will be keeping it separate. So you have two different type of layering here. One is called as the air layering. பப்பாயால யூஸ்வலாக இந்த லேயரிங் பண்ணுவாங்க. அந்த தேக்கு மரம் அதில் பண்ணுவாங்க இந்த லேயரிங். ஸோ இதில் என்ன பண்ணுறோம் அப்படின்னா here you can see that you are cutting a part of a stem here. ஸோ இந்த இடத்துல வந்து ஸ்டெம்மை வந்து கட் பண்ணிட்டோம். கட் பண்ணிவிட்டு யூ ஆர் ஜஸ்ட் ஆடிங் அ பிரான்ச் ஆஃப் அனதர் ஸ்டெம் அண்ட் இந்த இந்த இடத்துல வந்து அந்த அவுட்டர் லேயரை மட்டும் கட் பண்ணி எடுத்துட்டு அதாவது வெளியில் இருக்கிற அந்த பேரன்கைமா செல்ஸை மட்டும் கட் பண்ணி எடுத்துட்டு பார்க் அதை கட் பண்ணி எடுத்துட்டு யூ வில் பி ஆடிங் சம் மாய்ஸ்ட் சாயில் ஈரமண் போட்டுட்டு ஒரு கவர் போட்டு அப்படியே ஜஸ்ட் கட்டிடுவாங்க இந்த மாதிரி. ஸோ வாட் ஹேப்பன் ரூட்ஸ் வில் டெவலப் ஃப்ரம் திஸ் ரீஜன். இங்கே நீங்கள் பார்க்க முடியும்னு நினைக்கிறேன். இந்த கவரில் ரூட்ஸ் ஆர் ஆல் ஃபார்ம்டா ரைட். ஸோ அப்புறம் அந்த ஸ்டெம்மை தனியாக கட் பண்ணிவிட்டு தே வில் பி பிளேஸ்ட் ஆன் த சாயில். ஸோ such a very easy method of propagation. So just the outer layer is being peeled off. They will be covered with a moist soil and covered up and until they forms a root. After the roots have been formed, we just cut the stem along with the root. We will be placing on a soil. That's it. Fine, easy method, right? And the second one is called as a mound layering. As I said, jasmine. We are just bending up the branches and putting it in soil and we are placing up some weight over that. Let them form this root and then we will be cutting and placing it in a different region. So this is about the conventional method. So what is the advantage of the conventional method? Genetically uniform offsprings are being raised up by this method of conventional method. Genetically uniform. So here you cannot expect any difference in the plant. So if you are growing up a certain variety of plant in a very larger scale, so ஒரு பெரிய அக்ரிகல்ச்சர் ஃபீல்ட் பிரகாரம் ஒரே ஒரு வெரைட்டி ஆஃப் பிளான்ட்டை நீங்கள் வளர்க்கணும்னு டிசைட் பண்ணீங்க அப்படின்னா தென் திஸ் இஸ் அ மெத்தட். இப்போ நம்ம போயிட்டு செடியெல்லாம் வந்து கார்டனில் வாங்கிட்டு வரோம் இல்லை வந்து நர்சரிஸில் வாங்கிட்டு வரோம்னா they are using this genetically conventional method to get a genetically uniform breeds. Because இங்கே இந்தியாவுக்குள்ளே கிடைக்கிற ப்ரீட்ஸை மட்டுமே நர்சரிஸில் வளர்க்குறது இல்லை. நிறைய இந்த மாதிரி கட்டிங் கிராஃப்டிங் லேயரிங் பண்ண different varieties they are just making up and that should be followed up in all the plants of what they are selling. So this genetically uniform can be get through the conventional method and very quick. So need not to wait for the plants to do it on their own for a very longer duration of time will be required for it. So it is very quick a method to take up this as a vegetative propagation. And as you know that the plant is not going to produce any seeds that in case ஒருவேளை பிளான்ட் வந்து ரொம்ப கம்மியான சீட்ஸ் தான் ப்ரொடியூஸ் பண்ணுது, அந்த பிளான்ட்டை வச்சுட்டு ஒரு பெரிய அக்ரிகல்ச்சர் லேண்ட்ல சீட்ஸை வச்சுட்டு வளர்க்க முடியாதுன்ற பட்சத்தில் then you can go ahead for this conventional method, very easier and faster. Especially you can prefer this as a main method of propagation if the plant is not producing enough amount of seeds. And economically, this will be a good method, especially if you are taking for potato, Solanum tuberosum. This method is a very desirable method, and this is also gives you disease resistant plants and high yield. So because நான் சொன்னல genetically uniform அப்படின்னு சொல்லும்போது, so all varieties will be disease resistant. So money which is being spent for the pesticides can be saved if you are using this type of vegetative propagation method. That is especially the conventional method, short duration of time. So what is the disadvantage of using this? So virus infected plant, ஒருவேளை suppose நம்ம எடுத்துக்கிற அந்த mother plant, if it is being virus infected plant, then what happen? It will be followed to all the plants which you are propagating from that mother plant. So that is one disadvantage. And this vegetative structure, when you are propagating in a bulky form, கொஞ்சம் டிஃபிகல்ட்டாக இருக்கும் ஹேண்டில் பண்ணுறதுக்கும் ஸ்டோர் பண்ணுறது, பிகாஸ் ஹியூமன் மேனுவல் பவர் இஸ் ஹைலி நீடட். நீங்கள் போயிட்டு ஒவ்வொரு பிளான்ட்டையும் கட் பண்ணி ரூட் பர வச்சு அப்புறம் அதை எடுத்து சாயிலில் வச்சு, ஸோ இட்ஸ் ரெக்வயர் such a storage space, and also it requires handling of greater human power is required. That is the only small disadvantage on the conventional method. So humans are doing it. So what we are doing, we just want to make it in a very bigger level. We just want to need the help of modern methods. So it is a professor named F.C. Stewart, 1932, of Cornell University. He is the first scientist who just implemented modern technologies in vegetative propagation method in just to avoid this short duration and all these disadvantages are being controlled up. And he had given varieties of plants just in a laboratory. ஒரு laboratory ஒரு சின்ன ஸ்பேஸ்க்குள்ள ஒரு ஹைலி எதிர்பார்க்காத அளவு காம்பினேஷன் ஆஃப் பிளான்ஸை ஃபர்ஸ்ட் கொடுத்தவர் வந்து ப்ரொஃபஸர் எஃப்சி ஸ்டீவர்ட், கார்னல் யூனிவர்சிட்டியில். ஹி இஸ் த ஒன் ஹூ ஒர்க்ட் ஆன் த மெச்சூர் ப்ளோயம் பேரன்கைமா செல்ஸ். கேரட்லேருந்து எடுத்த பேரன்கைமா செல்ஸை ஒரு ஷூட்டபிள் மீடியத்தில் வளர்த்து வெறும் ஒரு டிஷ்யூஸ்லேருந்து கேரட் பிளான்ஸை வந்து டெவலப் பண்ணிட்டார். ஸோ தட் இஸ் அ மாடர்ன் மெத்தட் which is being used. How it is possible? Simply by the word called as toteopotency. The word toteopotency refers to the ability, the genetic ability of the plants. And the plants நீங்க எங்க இருந்து வேணா எடுக்கலாம். You can take it from a stem, leaf, ஒரு சின்ன cells இருந்தா போதும், a mass of cell. If you are putting them in an artificial medium, ஒரு laboratoryல petri dishல ஒரு மீடியத்துல, you can just put it up and they will form a mass of tissue and a small plantlet will arise, and the plantlet அப்படியே எடுத்து நேரா ஃபீல்டில் வச்சு. So lab is going to give you 10 by 10 room of lab is going to propagate it. Highly பெரிய டெக்னாலஜிஸ் இல்லை, சிம்பிள் டெக்னாலஜி டிஷ்யூ கல்ச்சர் யூஸ் பண்ணி you can able to do it. Many wonders can be done using this propagation methods. So it is called as a micro propagation method. Whole plant is developed from a very single cells or tissue. ஒரு சின்ன bit of plant இருந்தா போதும் உங்களால ஒரு full plant வளர்த்துற முடியும். So that is called as the micro propagation technique. And நீங்க நினைக்கிற மாதிரி எல்லா ஹைபிரிடிங்கும் இதுல பண்ணிட முடியும். ஆப்பிள் எடுத்து டொமேட்டோவோட மிக்ஸ் பண்ண முடியும், டொமேட்டோவை எடுத்து பொட்டேட்டோவோட மிக்ஸ் பண்ண முடியும். So all these wonders of hybridization can be done in the micro propagation technique. Very rapid technique where only lab is required, and labல பண்றதால ரொம்ப controlled நம்ம கிட்ட இருக்கும். So you cannot show any failures, எல்லாமே சக்சஸ் ஆகும். So that is a control will be with you peoples. And இது வந்து சீசனல் கிடையாது, கண்டினியூஸா வருஷம் ஃபுல்லா உங்களால இதை ப்ராபகேட் பண்ண முடியும் அண்ட் டிசீஸ் ஃப்ரீ ப்ராபகேட்ஸை உருவாக்க முடியும். கன்வென்ஷனல் மெத்தடோட பெரிய டிஸ்அட்வான்டேஜஸ், disease will be formed from the parent to the offspring. But micro propagation பண்ணும்போது sterile environmentல பண்ணும்போது you don't find such a disease in the propagules. So disease free propagules can be produced. ஸோ இன்எக்ஸ்பென்சிவ் ஒரு பிளான்ட்டுன்னு எடுக்கும்போது ஒரு லாட்ல நீங்க பண்ணும்போது பெரிய எக்ஸ்பென்ஸ் இதுக்கு செலவாக போறது கிடையாது. ஸோ யூ கேன் டூ இட் இன் அ லெபார்ட்டரி. ஸோ தட் இஸ் தி அட்வான்டேஜஸ் ஆஃப் modern method. Short duration, genetically identical offsprings can be produced. No need wait for any seasons and endangered. அழிய போற இனத்தை கூட நம்மளால என்ன பண்ண முடியும்? We can grow it up and we can save the species. அந்த species அழியாம பார்த்துக்க முடியும். And you can produce a viable seeds and meristem. Here you can produce a disease-free plants, and if you want, you can do any number of genetical modifications. So that is the high advantages of modern method. So what is the disadvantage? The simple disadvantage is that you require highly skilled persons to work here. So labor, that is a one. And you have to maintain the sterility in the laboratory. ஸோ ஒரு சின்ன பாக்டீரியா வைரஸ் எதுவுமே வராத மாதிரி ஸ்டெரைலாக வச்சுக்கணும் லேப்ல. ஸோ தட் இஸ் ஒன். ஃபார் தட் யூ ஹவ் டு ஸ்பெண்ட் சம் மனி. அண்ட் ஆஸ் யூ ஆர் ப்ரொடியூசிங் ஜெனட்டிக்கலி ஐடென்டிக்கல், யூ ஷுட் பி வெரி அவேர் அபவுட் வாட் இஸ் த ஸ்பீஷஸ் யூ ஆர் சூஸிங் ஃபார். அது நீங்கள் ரொம்ப தெளிவாக இருக்கணும். And then callus is actually a group of a mass of cell that is being formed on a petri dish is called as a callus. ஸோ இதில் எந்த அன்டிசைரபிள் சேஞ்சஸும் நடக்காத மாதிரி பர்ஃபெக்டாக நீங்கள் மெயின்டைன் பண்ணணும். இல்லைன்னா கமர்ஷியலுக்கு வரும்போது தே ஷோ சம் எரர்ஸ். அவ்வளோதான், சின்ன சின்ன டிஸ்அட்வான்டேஜஸ் தான். பட் டெக்னிக்கல் பர்சன்ஸ் அதை ஹேண்டில் பண்ணும்போது இட் வில் பி த வெல் அண்ட் குட் மெத்தட் டு டூ த ப்ராபகேஷன். ஸோ நேச்சர் லாங்கர் டியூரேஷன் ஆஃப் டைம் எடுத்து பண்ணுற விஷயத்த ரொம்ப ஷார்ட்டர் டியூரேஷன் ஆஃப் டைமில் ஒரு லேப்குள்ளே we can do it if you are skilled enough. So that's all about the asexual reproduction in plants. So I just wind up with this. So we talked about the vegetative propagation method in this natural, artificial, under natural, root, stem and leaf. What are all the examples அதெல்லாம் பார்த்தோம். நேச்சுரலோட அட்வான்டேஜ் டிஸ்அட்வான்டேஜ் டிஸ்கஸ் பண்ணோம். தென் ஆர்டிஃபிஷியல் மெத்தட்ல we talked about the conventional method and modern method. அதோட அட்வான்டேஜ் அண்ட் டிஸ்அட்வான்டேஜ் டிஸ்கஸ் பண்ணியிருக்கோம். ஸோ தென் செக்ஷன் 2ல we will be discussing on the செக்ஷுவல் ரீப்ரொடக்ஷன் அண்ட் பிளான்ஸை பற்றி டீட்டெயிலாக நம்ம பார்க்கலாம். தேங்க்யூ. உங்களுக்கு ஏதாவது டவுட்ஸ் இருந்துச்சு, இல்லை மேம் இந்த மாதிரி நடத்த நல்லாயிருக்கும், அந்த மாதிரி எந்த கமெண்ட்ஸாக இருந்தாலும் ஐ ஹைலி வெல்கம். அண்ட் ப்ளீஸ் புட் இன் கமெண்ட் ஃபார் இம்ப்ரூவிங் மை செஷன் அலாட். அண்ட் ஐ வில் கிவ் யூ வாட் எவர் யூ வாண்ட் இன் அ வெரி பெஸ்ட் வே. உங்கள் எல்லாருக்கும் எல்லா விஷயமும் போகணும். தட் இஸ் அப்ஜெக்டிவ் ஆஃப் மை டீச்சிங். அண்ட் ஐ திங்க் யூ காட் இட். See you in the next section. Thank you.